Hello everyone and welcome to the new session of the lecture series, and today's topic is plant growth and development. And today we'll be discussing and solving all the NEET previous year questions from this chapter. Okay, and we've already discussed some of the PYQs in the previous lecture, and today we'll be starting with the rest of the questions. So let's start with the first question, which is, so here they are saying the phenomena which is influenced by auxin, okay, and also play a major role in its discovery, means discovery of auxin. Okay, we have to mention the phenomena. We have to select the phenomena, and options are: first is phototropism, second option is root initiation, third is gravitopism, and fourth is apical dominance. Okay, so let's talk about the discovery of auxin first, or let's talk about the phenomena of phototropism. Okay, you can say, okay, phototropism or discovery of auxin, let us study. Okay, so Charles Darwin and his son Francis Darwin. Okay, so what they did, they were working on Charles Darwin and his son Francis Darwin. They were working on what? They were working on canary grass coleoptile. Alright. So what they did, they demonstrated the phenomena of phototropism. What is phototropism? Movement towards light. Okay. Now, what causes this movement? Okay. So for this, Charles Darwin and Francis Darwin did an experiment with canary grass coleoptile. Alright. So what they did initially, if this is suppose a canary grass coleoptile, okay, suppose this is a canary grass coleoptile. So what they did initially, if you illuminate, okay, it with the unilateral light source, so what is observed? Bending is observed. Okay, bending of the shoot tip or the coleoptile is observed towards the light. Alright. So Charles Darwin and Francis Darwin concluded that there is some substance which is causing this bending. Okay, there is some substance. Okay, they didn't say it is auxin, but they said there is some substance which is causing this bending initially. Then what happened? If suppose this is A, next what they did, they covered the tip of the coleoptile, okay, with a metal foil. Okay, again, if this is a canary grass coleoptile, they covered the tip here with a metal foil. They blindfolded it. Okay, now if they now illuminate the coleoptile with the light, okay, unilateral light, then no bending was observed. Okay, no bending of the coleoptile or the shoot tip was observed. Okay, then later again, if the light is allowed to pass through this foil, okay, or means you can say that if a transparent substance is applied instead of the metal foil, then what happened? Again, bending was observed. Okay, from this they concluded that there is some substance, okay, which is causing the bending of the light. Okay, and this bending of the coleoptile or the shoot tip is known as phototropism. Okay, later F.W. Wendt, okay, he was working on the Avena coleoptile. He isolated auxins, okay, which was IAA, indole acetic acid, which is a natural auxin. Okay, so what should be the correct answer? Answer is option A, phototropism. Alright. So now second option, root initiation, is not correct. Although auxins are involved in root initiation, but it does not. Okay. Second, gravitopism, again not correct answer. Apical dominance, again auxins are involved. Okay, but it is not the correct answer because phototropism led to the discovery of auxin and not the rest of the options. The next question is, which one of the following growth regulators is known as the stress hormone? Okay. Options are abscisic acid, ethylene, gibberellic acid, and indole acetic acid. Okay. Abscisic acid, also known as ABA. So let's study the function of ABA first. So abscisic acid, okay, is usually a growth inhibitor. Okay, it suppresses the growth. And what does it does? It promotes dormancy both in seed and the bud. Okay, seed you can write, you can say bud also, bud dormancy. What else it does? It helps to the plants to tolerate abiotic and biotic stress. Okay, now abiotic means environmental stress such as drought, okay, salinity, high salt condition. Biotic stress caused by the microorganisms, or you can say pathogens. All right. So these abiotic and biotic stress, okay, ABA helps to tolerate them. So that's why ABA is also known as the stress hormone. It is produced during the stress. Okay. So option A is abscisic acid, and they are asking in the question about the stress hormone. So option A should be the correct answer. Next option is ethylene. Ethylene is known as the ripening hormone. Okay, because it helps in the ripening of the unripe fruits such as bananas. Gibberellic acid. Okay, it is, what are the roles of gibberellic acid? Bolting, malting. Okay, promote the seed germination. Okay, it promotes seed germination. What else it do? It speed up the malting process. Okay. Second, it also induce bolting. Okay, helps to increase the internode length. Indole acetic acid, it is an auxin, natural auxin. Okay, and what are the function? It promotes apical dominance. Okay, it helps to induce parthenocarpy. All right, also helps to induce the roots in the callus. Okay, so correct option is option A, abscisic acid. Now third question is, we have to match list 1 with the list 2. Okay, so here is the list 1 and 2. So first we have the auxins. All right, so auxins, these are the options: promote fever flower, overcome apical dominance, increasing the length of the grape, and promote flowering in pineapple. Okay, so this should be the correct answer. So A is fourth option. Then let's talk about B, gibberellin, gibberellic acid. Increase in the length of grape stalk, and then we have the promote female flower in cucumber. So what should be the answer? Gibberellins usually help to increase the internode length. Okay, so increase in the length of grape stalk. So option B is third. Then cytokinin. Cytokinin helps to overcome the apical dominance and it promotes the lateral growth. Okay, cytokinin promotes lateral growth. All right, so option C should be 2. Now coming to the Last option, ethylene. Ethylene promotes female flower formation in cucumber, and this leads to the increase in the yield. Alright, so option D should be 1. Okay, now these are the options. You have to select from them. Okay, you can take your time. So what should be the answer? Answer is option B. We have already discussed how we achieved this option. A is 4, B is 3rd, C is 2nd, and D is 1. Alright. Okay, this was the option C. Now coming to the next question, which is which of the following statement is not correct? Okay, the first statement is phase of cell elongation, okay, of the plant cell is characterized by increased vacuolation. Alright, so let's talk about here they are talking about the phases of the growth. And there are 3 phases of growth. Okay, or growth takes place in 3 phases. First is the meristematic phase. During meristematic phase, what happens? Okay, the cells are rich in protoplasm. And they have conspicuous nuclei as well. And if we talk about the cell wall is primary and cellulosic and thin. Cell wall is primary, okay, thin and cellulosic. Alright, and there is lot of plasmodesmatal connections known as cytoplasmic bridges. Alright. Now let's talk about it. Next phase is the elongation phase. Okay, during this what happens? Cell enlargement takes place. Cell enlargement. And then what else we have? We have the increase in the number of vacuoles. Number of vacuoles means increased vacuolation. Alright, and what else? There is deposition of new cell wall material also. Okay, and then last phase is the maturation phase. During maturation phase, what happens? The cells have attained the maximum size. Okay, maximal size. Okay, in terms of what? Cell wall deposition and protoplasmic modification. Alright. So now, again coming back to the first option, phase of cell elongation of the plant cell is characterized by increased vacuolation. Okay, increased vacuolation. Okay, so this is correct. But we have to select the statement which is not correct. Then option B is cells in the meristematic phase of the growth exhibit abundant protoplasmic connections. Alright, lot of protoplasmic connection in the meristematic phase. So this is also correct. Option C is plant growth is generally determinate. Okay, and option B, last option is plant growth is measurable, which is also true. We can measure the plant growth. Okay, we can quantitatively measure the plant growth. And again coming back to option C, which is plant growth is generally determinated. So this is not correct because the plant growth is open type, or you can say it is indeterminate. Alright, means it is not terminated. Okay. So option C is the correct answer for this question. The next question is the ability of plant to follow different pathways in response to environment, leading to the formation of different kind of structures is called. Okay. So the options are differentiation, redifferentiation, development, and plasticity. All right. Differentiation, differentiation of the undifferentiated cell will lead to the formation of mature cells with specialized function and characteristic. Okay. But there is no formation of different kind of structures. All right. So this correct option is not correct. Second option is redifferentiation. All right. Redifferentiation, when the cells lose the capacity to divide again. To divide again. Okay. And then they become mature cells. This is known as redifferentiation. Now let's talk about development. Okay. Before redifferentiation, we have dedifferentiation. Dedifferentiation means when the cells again gain the capacity to divide. Okay. Then they become dedifferentiated cells. And then we have redifferentiation when they again lose the capacity to divide. Now, next option is development. Again, development means all the stages from the germination of the seed. Okay, if we talk about the plants, from germination of the seed to the senescence. Okay, and senescence ultimately leads to death of the organism. So this is also not true. Now, last option is plasticity. All right. Now, plasticity means when the same organism, all right, forms two different structures, okay, or show two different phenotypes. This is known as plasticity, or simply you can say ability to mold. Okay. Now, plasticity again can be of two types, environmental and developmental. Okay. So, what should be the correct answer? Answer should be option D. Plasticity is the phenomena where the plant follow different pathways, okay, in response to either the environment, okay, leading to the formation of different kind of structures. Now, next question is, which of the following growth regulator is an adenine derivative? So, growth regulator here means growth hormone. Okay, plant growth hormones are a type of plant growth regulators. All right. Options are abscisic acid, auxin, cytokinin, and ethylene. Okay. So, abscisic acid is a carotenoid derivative. Auxins are indole derivative. Cytokinin, these are the adenine derivative. Alright, that's why there is example nucleic acid also. Ethylene is a gaseous hormone. Okay, it is C2H4. Okay, so what should be the answer? Option C, cytokinin is an adenine derivative. The next question is, if you are given a tissue with its potential for differentiation in artificial culture, alright, which of the following pairs of hormones would you add to the medium to secure shoots as well as roots? Okay, so here they are saying, suppose there is a callus, which is the mass of undifferentiated cells. Callus is a mass of undifferentiated cells. Okay. So, which hormone should be given? Which hormone treatment should be given so that it should form either, okay, both shoots and roots. Alright, now we already know that ratio of auxin and cytokinase is responsible for either the initiation of shoot or root. Okay. Now, if there is a high auxin to cytokinin ratio, okay, high auxin to cytokinin ratio, then what happens? Roots are developed. If there is high cytokinin to auxin ratio, then shoots are developed. Okay. So in general, both auxin and cytokinin are required. So options are gibberellin and abscisic acid, which is not true. Abscisic acid is a growth inhibitor. Gibberellins have some other functions, okay, like seed germination and all. Next option is auxin and abscisic acid. Okay, so this is also not true. Abscisic acid again a growth inhibitor. Third option is auxin and cytokinin, and fourth option is IA, which is an auxin and gibberellin. So this is also not true. So what should be the correct answer? Auxin and cytokinin should be present in the medium, okay, for the development of both shoots and roots. The next question is Wendt, okay, FW Wendt noted that if the coleoptile tips were removed and placed on the agar, okay, for 1 hour, the agar would produce a bending when placed on one side of the freshly cut coleoptile stump. Okay, of what significance is this experiment? Okay, so what did Wendt did? He was working on the Avena coleoptile, oat coleoptile. Avena is oat. Alright. So what he did? If this is a Avena coleoptile, okay, so here in this region, in the shoot tip, what is produced? Auxins are produced here. Okay, this red region is the auxin. Then what happened? He cut off this shoot tip. Okay, so here, this is the coleoptile stump, okay, without the shoot tip. Coleoptile stump, it is called coleoptile stump. Okay, then this part, okay, which he removed, he kept it on an agar block. Okay, he kept it on the agar block. So initially auxin were present here in this shoot tip. Then this auxin diffuses into the agar. Okay, it diffuses into the agar block. Okay, then the next is what happens? The auxins reaches in the agar here from the shoot tip or the coleoptile. Then what happened? He kept this agar on the coleoptile stump. Okay, he kept it on one side here. And then what he did? He illuminated it with the unilateral light. So what happened next? There was bending was observed. Now again, although the shoot tip has been removed, it has been decapitated, but still bending was observed. Why? Because the auxin now, okay, auxins from the agar block now diffuses into the coleoptile stump. Okay, so this was the experiment done by Wendt. This is known as the agar block experiment. Alright. Now we have to select from the options what is the significance of this experiment. First is it demonstrates the polar movement of auxins. Next is it supports the hypothesis that IEA is auxin. Next option is it is the basis for the quantitative determination of small amount of growth promoting substance. And fourth option is it made possible the isolation and identification of the auxins. Alright, so let's discuss the first point. Polar movement of auxin. Okay, we already know that auxins, okay, show polarity or the polar movement. Okay, auxins. What is polar movement? Polar movement means directional movement in one direction. Okay, and auxins, okay, are produced in the shoot tips. So they move from the shoot towards the root. Okay, so this movement requires energy, means it is an active process. Okay, and it is directional, means it is polar. Okay, so first option is this. So now what should be the correct answer? Answer is option A. It demonstrated the polar movement of auxins. Alright, so what happened in the vent experiment, agar block experiment? Auxins from the shoot tip, okay, were transferred first to the agar block, and from the agar block again it was transferred to the coleoptile stump. So there is a directional movement from shoot towards the root. Okay, other options are not correct. Now next question is, a few normal seedlings of tomato were kept in dark. Okay, you can circle it. They were kept in dark, okay, room. After a few days, they were found to have become white colored. Okay, means they become pale. White colored means there was no chloroplast or there was chloroplast degradation. Okay, and they became albino, means white color. Albino means white or pale. Now, which of the following terms will be used to describe them? Okay, options are defoliated, etiolated, embolized, and mutated. Okay, so defoliation, what is defoliation? Okay, removal of, you can say, leaves. Okay, either purposely or naturally. Alright, then let's talk about embolize. Embolize is the presence of or appearance of bubbles, air bubbles or oxygen bubbles in the plant water transport system. So what happens? Usually a continuous column is required, but sometimes there is an air bubble in the plant water transport system, and this can inhibit the growth. Alright, and it can also inhibit the water uptake also. Okay, or decrease the water uptake. Now next option is etiolated. So what is etiolation? Turning of the plants to either pale, okay, or they become very weak. In dark, you can say, when they are grown in the dark, or you can say without light, when they are grown without the sunlight. So sunlight is very important for the plants. Without sunlight, there is not proper development of the plants. There is no chlorophyll, okay, generation and all. So plants become pale and very weak. So in the question also they are asking, they have become white colored when they grow in dark. Okay, so this should be the correct answer. And fourth option is mutated. They are mutated. Okay, which is not true. When the plants are kept in dark, they won't mutate. Okay, how can we define mutation? They are the genetic mutation, random genetic mutations. Okay. So what should be the correct answer? Answer is option B, etiolated. Okay. The next question is, the pineapple, which under natural condition is difficult to blossom, okay, has been made to produce fruits throughout the year by the application of, okay, options are naphthalene acetic acid and 2,4-D, 2,4-dichlorophenoxyacetic acid. Then we have option B, phenyl acetic acid. Option C is indole butyric acid and indole acetic acid, which are natural auxins. Okay, so these are natural auxins. These ones are synthetic auxins. Okay. And the last option is cytokinin. Now, what is cytokinin? Okay. It helps in the cell division and delay in the senescence. So here the correct option is answer is option A. Synthetic auxins, okay, are used, okay, so that the pineapple can blossom, okay, it can produce flowers. The next question is, which of the following generally acts as an antagonistic to gibberellins? Options are indole acetic acid, which is an auxin, then we have ABA, ethylene and zeatin. Okay, zeatin is again a cytokinin. It is a natural cytokinin. So what are the function of gibberellins? Gibberellins, okay, they promote seed germination. And what else they do? They also delay the senescence. They delay senescence. Okay. But whereas abscisic acid is the opposite of gibberellic acid. Abscisic acid, it promotes dormancy. It promotes abscission also. Okay, and it also promotes senescence. So it is an antagonistic to gibberellic acid. Its functions are antagonistic to gibberellic acid. Promotes senescence also. Alright, it promotes abscission also. Okay, whereas gibberellic acid acts opposite to ABA. So what should be the correct answer? ABA. Next question is, coiling of the garden pea tendrils around any support is an example of thermotaxis. Thermo means heat, means movement towards a heat source. Okay, movement towards heat. Okay, then we have thigmotaxis. Thigmo means touch. Okay, so you can say change in the direction of the locomotion, okay, because of a touch stimulus. Okay, change in direction of the stimulus, direction of locomotion, okay, due to a touch stimulus. Now what is thigmonasty? Thigmonasty is again related to the touch stimulus. Okay, but this is not directional. It is not directional. Okay, doesn't happen in one direction. It can happen in random direction. What is thigmotropism? Again related to touch, but the movement is directional. Directional movement. Okay, directional movement. So in the question they are asking, coiling of garden pea tendrils towards any support is example of option D, thigmotropism. The next question is, senescence is an active developmental cellular process in the growth and functioning of the flowering plants. So it is indicated in, okay, options are leaf abscission, vessels and trachea differentiation, floral plants and annual plants. Okay. So what is senescence? Senescence is also known as biological aging. Okay, and what happens during senescence? There is a loss of the function and the characteristic of the organism. Okay, it is a deterioration process. Function and characteristic of the cells or the organism. Alright, so first option is leaf abscission. Okay, means falling off of the leaf or detachment of the leaf. Okay, so in this what happened? An abscission zone is formed which limits the nutrients. Okay, nutrients cannot pass to the leaves and the leaves falls. So this happens during the senescence. Okay, in mature leaves. Leaves which are mature and old, they fall off. So it is a part of the senescence. Next option is vessel and trachea differentiation. Okay, means formation of mature cells. Vessels and tracheids, okay, these are mature cells formed by the process of differentiation. Then the third option is floral parts and annual parts. Again, this is not true. So what should be the correct answer? Leaf abscission. Next question is auxin can be bioassayed by. Okay, this we have already discussed. Options are Avena coleoptile curvature. Okay, here we have discussed the experiment done by the Wendt. Next is hydroponics. Hydroponics is growing of plants in water, or you can say nutrient solution, without the soil. Hydro means water. Next option is photometer. Now, what is a photometer? Okay, photometer can detect the electromagnetic radiation. Okay, it is for the detection of the electromagnetic radiation. Okay, from ultraviolet to the infrared, including the UV visible spectrum also. Then lattice hypocotyl elongation. This is a bioassay for gibberellins and not auxins. Okay, because gibberellins helps in the elongation of the internode, hypocotyl as well. So what should be the correct answer? Avena coleoptile curvature. The next question is typical growth curve in plants is sigmoidal, linear, shear-shaped and parabolic. Okay, so we know that we have already discussed when we were studying the chapter that a sigmoidal growth curve is observed during the which phase? Geometrical, during geometric growth or exponential growth. Okay, when one cell divides into two and the other two cells also have the capacity to divide. Now these both cells can divide into two as well as these also. So in this case, a sigmoidal growth curve is seen. This is a sigmoidal growth curve which is S-shaped. Here we have the lag phase during which the growth is very slow. Then we have the log phase or the exponential phase during which we see rapid growth. Okay. Then we have the stationary phase in which the growth decreases due to the depletion of the nutrients. The growth decreases. Alright. Here we have the time. Here suppose we have the length of the organ, or you can say height of the plant. Alright, so typically a sigmoidal growth curve is observed. Okay, when we talk about the growth in the plants. Linear growth curve, okay, example can be elongation of the root. Okay, alright, or you can say during the arithmetic growth rate. Okay, the other options are not correct. Okay, so correct option is sigmoid. Alright, so this was the end of this lecture. Thank you very much for listening.